it is, I think, perfectly fitting to honor someone like Sam, who's had such a profound impact on psychiatry, that we have managed to get Karl Deisseroth here as the first Barondes lecturer. So if you look on your programs, you can see several paragraphs summarizing Karl's biography, his professional biography, and it is a breathtaking one, from National Academy of Medicine, National Academy of Science, Howard Hughes Investigator, to the Breakthrough Prize. And I would say that as scientists, we all tend to gripe about the overemphasis on publishing stats and high-profile journals, but I'd also venture a guess that every scientist in the room would turn quickly to the bibliography of anyone that they happen to meet in order to get a sense of the quality and impact of their science. And in this regard, Karl's numbers, as it were, are staggering. Over 250 publications, but numbers are not everything. I have to say, however, that when you take a look, 88 of them have already been cited more than 100 times, which I think is a remarkable testament to the impact that the work has had in really less than 20 years. When you take a look, nearly every publication, dishearteningly, is published either in Nature, Science, Cell, or Neuron. It's an extraordinary record of achievement, but the honors and awards, fundings, and stats really don't capture what I'd like to try to convey for those of you who are not familiar with Karl's work. But he is truly one of the rare scientists that has, I think, unequivocally transformed a field, an entire field of inquiry. And his impact on psychiatry, as Sam has just mentioned, is just beginning to be felt. His discovery and implementation of optogenetics, which allows neuroscientists to manipulate brain circuits in living model organisms, has truly revolutionized our field. And if that wasn't enough, he followed this up with the development of a tool called Clarity, which allows us to visualize and study at the molecular level the post-mortem brain while retaining its three-dimensional structure. So this is a true story. I was down at Salk Institute several years ago to participate in a symposium on progress in psychiatry and neurodevelopmental disorders. And there was a group of very, very successful investigators in the lineup. Karl was among them. When Karl gave his talk, I was sitting in a row of all of the speakers. And Karl introduced us. It was the first time I, or I think anyone in the row, had seen Clarity. And he had an image that had us flying through the three-dimensional neuronal structure of human brain. And I looked down the row at all the other investigators, and the looks on their faces just reminded me, the only thing that came to mind was the scene in Wayne's World, where they drop to the floor and say, "We are not worthy." You recall that to Alice Cooper? So you're the Alice Cooper of neuroscience. And I have to say, at that moment, I was simply thanking my lucky stars that I was not the next person in the lineup. So, the choice of Karl as the first Barondes lecturer was a no-brainer, but especially fitting for a couple of other reasons. First, he truly is the apotheosis of the physician-scientist. Despite all of the accolades, all of the awards, and all of the progress in his laboratory, he still finds time to see patients and to supervise residents in a treatment refractory mood disorder clinic. I think is a remarkably wonderful thing. And he's also set the bar not only as a scientist, but as a mentor. The first author, when you read through the list of his publications, reads like an ever-expanding list of scientific superstars, including, fortunately for us at UCSF, our own Vika Sohal and Lisa Ganaden. So welcome, Karl. Thank you so much on behalf of Sam, on behalf of the selection committee, the audience today, and myself for accepting the invitation to be the inaugural lecturer in the Barondes Lectureship.
Thank you, Matt and Sam. I'm not worthy. I have so many friends and colleagues, people who've worked with me, people I've admired for so long. This is for me, it's a very special moment, and I'll tell you a little bit more. I might take a few minutes even now to try to give a sense of what Sam and his work have meant for me, not just now, but in the past as well. I've known Sam for a long time. I've been influenced by his work for many, many years. But I also would like to roast him a little bit, if that's okay.
Be my guest.
Okay. Now, I don't often, although my lab is a basic science lab, but I'm continually inspired by psychiatry and my patients, and I have rare opportunities though to actually talk about that. And so this, what I'll talk about today, there's some very exciting unpublished work that I'll spend most of the time on, all related, although it's basic science in one way or another, to psychiatry. And so I'm looking forward to that as well. It's a bit of a special moment in that regard. Now I, like many people in the room, I want to keep things up to date, up to the minute, so this is an iPhone picture from the program. And a few things, there are various things in Sam's background that have meant a lot to me that people may not know about. He gave a very powerful depiction of his work on the genetic code and also his identification of the importance of protein synthesis for memory. In my PhD work, which nobody remembers now, but seemed important at the time, we did a lot of work on understanding the coupling between neural signals and gene expression. And this work, in many ways, built on the foundation that Sam laid. Also, many other aspects of what Sam has done have meant a lot to me. Of course, his work with the McKnight program, which supported me quite a bit early on, was very important. Finally, I'll come back to the other aspects, including the genetic code. But what's amazed me is his appreciation and action on behalf of art and literature. A lot of people, appreciate literature, appreciate the beauty of what comes from your artistic temperament, but Sam actually delivers to and generates it himself, which I think is amazing. And I appreciate poetry. Many of you may know that his most recent work is actually poetry, and I've delved into what I think are some of his influences. And one of them, I think, is he may not know this, but one of them is Emily Dickinson, who was one of my favorite poets from before the last century. Now, I went and found a poem of Emily's and a poem of Sam's, and I put them up side by side. Now, Emily, as some of you may know, she tips her hand a little bit. She uses dashes and capitalizations that would give you a clue. So I've taken the liberty, Sam, of adding some dashes and capitalizations to your poem as well to make it more difficult to figure out. So one of these is from Sam, and one of these is from Emily Dickinson. There is a dial inside my brain that turns to make me sad. It sits beside another dial that turns to make me glad. I'd like to learn to grab them, to turn them up or down, to give myself a joyful smile and take them away. But every time I reach in, the dials just seem to hide. I've never learned to find them. My brain's too dark inside. Powerful stuff. The brain is wider than the sky, for put them side by side, the one the other will contain with ease, and you beside. The brain is deeper than the sea, for hold them blue to blue, the one the other will absorb, as sponges do. The brain is just the weight of God, for heft them pound for pound, and they will differ if they do, as syllable from sound. Now, these are similar structures, as you can see. We've got 3 stanzas, 4 lines each. Setting up the problem with the first 2 stanzas, and then coming in with powerful sensory system metaphors at the end. Powerful lines at the end. So, many of you may have some thoughts on which is Sam, which is I'll just tell you this is from Sam's, of course, book for children who think, as he so beautifully put it. But this also is nice because it captures what's, I think, always made me feel close to Sam, is his lack of pretension coupled with his creativity and brilliance. He's genuine, straight shooter, just a pleasure to talk to always. And as you can see from his career, his ideas have been so far ahead of his time, often too far ahead of his time, and that's been something that you can imagine is not an easy life to lead, to be too far ahead. And I have tremendous respect and love for what you've done. So it's an honor to be here to give this lectureship. And I want to share work that does relate to the in some ways the vision and spirit of Sam. Now, a lot of the work that I'll talk about is unpublished, and a lot of it does depend on another theme, which is molecular approaches to neuroscience and biology. And even, of course, I'll tell you about some of our work in trying to understand some of the beautiful proteins that come from nature that have given us some toehold on some of these complex patterns. And of course, without the pioneering work that dated way back to his work with Marshall Nuremberg, we wouldn't be able to do this either. But even more yellowed with age than that book that he wrote is some early efforts on trying to describe back in 1809 the symptoms of madness and melancholy. And here I take inspiration from looking at this treatise by John Haslam in 1809, who described what we might call the anhedonic symptom of depression or melancholy. And his words were, and this is before, by the way, the nosology of Kraepelin and colleagues who organized psychiatry into a form that took on a medical model, but Haslam used medical model language for these complex problems, and he said, regarding the patients with melancholy, that they therefore neglect those objects and pursuits which formerly proved sources of delight and instruction. And this is both beautiful and true, but also depressing, because our ability to describe and diagnose anhedonia or depression in general essentially is like this now. We don't have a lab test or a deeply empirically based quantitative measure to understand or to diagnose or quantify this powerful and important symptom. And so this is something that has needed a new approach, a new way of giving us a way of interacting with the brain in a fashion that gives us deep understanding of circuitry. And what's been an amazing story is that proteins from microorganisms, algae and archaeobacteria, and in particular the amazing properties of these proteins, it's this that has given us a toehold that has allowed us to understand the symptom of anhedonia and other symptoms of psychiatric disease. In addition, of course, to their basic science role, which is the main function of lab that I run and that most people who use these tools operate with. I want to tell you about the proteins and our understanding of how they work and how they're used. I'll get to that in the second half. I'm going to start with unpublished work that is also very psychiatry inspired and potentially relevant, but from another very small system, not as small as the single-celled organisms, but larval zebrafish, which give us some unique advantages. And we're still working on these papers and writing them up, so this is really hot off the pre-presses, not even presses yet. And so let me set the stage a little bit. As many of you will know, we have in neuroscience and in clinical medicine interventions that are electrical or magnetic, that are fast and targetable spatially, but they cannot resolve individual cells that are right next to each other, that may have completely different roles. The essence of the optogenetic approach is to introduce these proteins from microorganisms, the microbial opsins, which are single component, single gene light activated regulators of ionic conductance. And these respond to different colors of light, let's say yellow or blue, and pass different ions of different kinds. And one can use these, as we found, to turn neurons on or off, and to do this in freely behaving mammals carrying out complex tasks. And I won't talk too much about it, but I'll just give some very brief examples of that to make sure everybody in the room is up to speed on the nature of the method. So we can deliver the genes themselves, the light-activated regulators of ion flow, via viruses. We can inject them into a brain region like the basolateral amygdala or the bed nucleus of the stria terminalis. And we can then deliver light via fiber optic deep in the brain, either to that region or to downstream regions. And the value of that approach is you can recruit cells that are defined by origin and target. The cell bodies are here. The microbial opsin is shipped within the cell down the axons, and you can recruit the cell defined by its connectivity, which is a very powerful approach. And this movie shows an example of this in the symptom domain of anxiety. This is a mouse in an elevated plus maze. The elevated walls of the maze create the closed arm. That mouse much prefers to spend its time in the closed arm of the maze. It very rarely ventures into the open arms, which are exposed. And so this is apprehension in the absence of immediate threat or anxiety. And when the blue letters come on here, that's when Kay Tai and other colleagues in the lab did an experiment controlling, activating at the projection within the amygdala, defined by origin and target, that turned out to be anxiolytic. For the first time, the animal is willing to include the open arms in its exploration. Nothing else changes about the animal. It still explores the closed arms. Its speed doesn't change, but it's instantaneously willing to go into the open arms. And this is reversible. When you turn off the light, you stop controlling this projection within the amygdala, the effect disappears. And this is an example of a causal understanding of what a cell population defined by its anatomy can do. Now, of course, this is only one aspect of anxiety, which is a risk avoidance feature. We know anxiety is complicated, it has other features as well. There are physiological changes, heart rate changes, and respiratory rate changes. And then there's a subjective balance to it. It's aversive, it has negative balance, and the resolution of anxiety is a positive balance. It's appetitive. And only one thing is being measured here, which is the risk avoidance. But in subsequent work from other folks in the lab, we found that there are different projections within the brain that recruit different features of the anxious state. The respiratory feature is recruited by a projection from the BNST to the parabrachial nucleus in the brainstem. The risk avoidance feature is largely mediated by a projection from the BNST to the lateral hypothalamus. What's very interesting is this, and this is quite, I think, profound and surprising. Even though there's a powerful risk avoidance effect mediated by this projection from the BNST to the lateral hypothalamus, there's no appetitive or aversive quality to it for the animal. The animal doesn't care that it's happening, which is very interesting. It shows the separability of the valence of the state from the complex behavioral modification that is elicited. And that separability is very intriguing. Where the valence comes from, why the valence is attached to certain states is, needless to say, of profound importance, not just for psychiatry, but for neuroscience. And that valence of the state seemed to be mediated by a projection to the ventral tegmental area. And we found recurrent circuitry within the BNST that might help to coordinate these different projections, which go out and recruit different features of the complex state and may make them come on together and go away together. So this example illustrates how complex behavioral states of natural significance and of relevance to psychiatry can be studied with this approach. Now, what I want to talk about today is unpublished work on a different class of disorders. Again, natural and adaptive in some sense, but also one of the behavioral states that can become maladaptive. And this is also relevant to my particular clinical specialty, which is treatment-resistant depression. Depression, as you know, is definable by a number of different symptoms, and among these are some that we can measure in animals. So we don't claim in any of the work that I do to be recapitulating a human disorder, but we're interested in the behavioral states and the transitions among the states, how they're elicited, how they're suppressed in natural behavior and in states that are stress related. Depression has some prominent features, including passive quality to it, hopelessness in people. You could define hopelessness as a deep discounting of the value of action in the present. And this, as we know, is of profound significance for depression, for patients, for families, for their occupations, for their overall functioning. And this passivity or passive coping with challenge is something that can be readily studied in animals. There are ways you can address challenges with an active coping strategy, and then there are passive coping strategies, and those may both be appropriate under different circumstances. But a too ready or too powerful transition to the passive state can be pathological and often is. And so that we think of as hopelessness in people, we refer to it as passive coping in There's also prominent anhedonic features to depression. Such an important feature of depression, the inability to feel reward or to be motivated by pleasure or reward or interest. This is such an important part of depression that you can actually be diagnosed with depression without having depressed mood if you also have anhedonia. This is a very fundamental feature. And different people may express different classes of these symptoms. Now, back in 2012, Melissa Warden in the lab did some experiments wherein she studied using the well-established and well-validated forced to swim test with rodents. And this is a challenge which the animal meets with first an active coping strategy and then a transition to a passive coping strategy. The active coping strategy is seeking to escape, climbing, swimming, and passivity is simply floating. And these are this transition from active to passive is an important transition to understand. And what Melissa's advance in this case was to integrate this complex task with fiber optics for controlling particular projections, and she had a tetrode so she could record from prefrontal cortex during this action, and she could pick up the movement of each little kick of the animal with a magnetic coil readout. And this allowed us to study with very high resolution in a causal fashion this transition from active coping during the early stage of the test to a passive state later. And so this is, you can look at it from the frame of an adaptive behavior, or if this is of interest, you can think about it in terms of maladaptive behavior. So it's a natural, these are rats that are not in any sort of stress state. They're not a disease model. These are wild type rats. Now, this has been studied by many people over the years. The basic brain region related foundations of the field has pointed to a number of different structures across the brain that, when lesioned or modified or pharmacologically affected, can influence behavior in this test. And what Melissa was interested in was understanding, are there projections, connections from one part of the brain to another that mediate this behavioral state transition, which was an inaccessible bit of knowledge until the advent of optogenetics. And she found some very interesting things. For example, she injected a channel rhodopsin, an excitatory microbial opsin that's recruited by blue light, into medial prefrontal cortex of rats, but she delivered light to different downstream structures. For example, the lateral alpha, when she drove cells defined by origin in prefrontal and target in lateral alpha, she saw a favoring of the passive coping strategy. So here you can see the kick frequency. Each blue epoch is when light is delivered, and you can see a modest but reliable and reversible favoring of the passive coping strategy every time she drove cells defined by this. Not seen if you're just expressing a yellow fluorescent protein. This was not seen in contexts that were not challenging like this. In the open field test, there was no effect on locomotor behavior, for example. And other projections didn't have this effect. Projection from prefrontal to basolateral amygdala, for example, had no effect, and another projection had the opposite effect. And this was the projection to the dorsal raphe nucleus. And here, every time she drove cells from prefrontal to dorsal raphe, she was able to favor the active coping strategy. And again, this was not seen when just expressing a yellow fluorescent protein, and also not seen in the open field test. No non-specific effect on animal behavior. So this was very interesting that within prefrontal cortex, which many reasons to think this is, at least to some extent, the site of a generation of complex motor plans and estimation of the value of action in the present, at least with regard to possible consequences in the future, that different projections coming from prefrontal cortex can recruit and favor the active or the passive coping strategy. So this was very interesting. At the same time, it was also, we were aware of the limitations of this work as well. In all of these experiments, one has to carry out very targeted experiments. You have to have a specific hypothesis, which is fine, nothing wrong with hypotheses, but you have to think, I'm going to test this projection to this target, and in some respects, it's like the parable of the drunk individual looking for his keys, but only looking under the street light because that's where the light is. In some ways, we were looking, because of the nature of the adult mammalian system, looking only under the street light. So the new work I want to tell you about is one of our efforts to get beyond that situation and take a truly global, brain-wide, cellular resolution real-time analysis. And what we've done is to go to the larval zebrafish. Now this animal is a vertebrate, of course, has a vertebrate body plan, a vertebrate brain plan, and there are, as you know, this is now a widely used model animal system in many fields of science, ranging from developmental biology to cardiology to neuroscience. And the unique advantages of this, in this case, is that we can see the activity of every single neuron in the forebrain in real time. And this was not known until the two talented individuals in the lab, Aaron Andelman and Vanessa Barondes, did their work. You can actually find a behavioral state transition from active coping to passive coping under conditions where you can see the activity of every single neuron in the brain. And so this was the value of this approach. Now, how could you do this? This required some infrastructure, some setup. These are beautiful tanks, but nobody cares about that. I like them, but they're I'll just move on from that. But here's another challenge. You can't really do a forced swim test with fish, right? I really like maybe there might have been ways around that. Forced not swimming, maybe that could. But what Aaron and Vanessa found was a paradigm where, under conditions in this state of the animal where we can see the activity of every neuron, they acted to passive behavioral state transition that had the required properties. And this took a number of years to sort out. The way they did it was this is a little bit like this is a very simple apparatus. It's just a tank, and I'll show you some modified versions of this a little later. But an animal, if the first half of the swim period that you're measuring is coded in purple and the second half in orange, the fish will keep swimming in the first half and the second half over many tens of minutes without a change. But if halfway through you administer a mild, inescapable aversive stimulus, a very mild 1 Hertz electrical shock delivered across the tank, the animals have this very interesting transition. They stop moving as much. They enter into a passive state. And like the rodent in the forced swim test, there's first an active coping phase and then a transition to a passive coping phase. So that was very interesting. We were interested in seeing that for validity relative to the mammal. And it had other interesting properties as well. You may say, well, you're just cooking the fish and then you should have a dead fish at that point. But it's not the case. We've set this up to be very viable, but also the fish recover completely when you put them back into a new tank with new water. They return to completely normal function. But this is in a context-dependent way. So it's not, but in a way that shows that there's memory and that there's a context-dependent memory. Now, it takes some tens of minutes for them to return to the active state. So it's not like a contextual fear response in a mouse where if they've experienced an aversive stimulus in one chamber and they freeze, you move them to a new context, they immediately start moving around again. That's not the case here. There's a persistence to the state, which lasts some tens of minutes, and actually even longer, as I'll tell you about later. Also, it's context dependent. If you keep them in the tank where all this happened to them, they don't change their behavior. It's shown in green, so they maintain their passive state. And if you change either the tank or the water, these are the partially neutral environments, you get an intermediate effect as shown in yellow. It was also pharmacologically modulatable, and so here we needed a fast-acting agent. Many of you may know there's interest in ketamine as a possible fast-acting agent. We don't really know what this means, but we certainly do know that ketamine, when administered 1 hour before the start of this protocol of these fish, powerfully slowed the transition to the passive coping state. And we actually had to extend, this would be the normal termination of the protocol, we actually had to extend the protocol to see that it doesn't completely block the state. The animals will eventually enter into the passive state. So it's not a complete effect, but it shows that this transition is pharmacologically modulatable as well. So this behavioral state transition had a lot of the properties that we thought were interesting, and all of this, by the way, this was our requirement. The task I set to Aaron and Vanessa was we have to make sure that this transition happens at a stage where we can see with single cell resolution the activity of every neuron in the brain. And it wasn't known that you could have this at this particular stage of development. Now, so what is going on in these animals during this transition? There are homologous structures in the fish, and then there are structures which they lack. They don't have a proper neocortex, as you know. They do have raphe nuclei. They have habenular structures that are homologous to our and to the mammalian lateral and medial habenula. The nomenclature is a little different. I'll use the mammalian nomenclature here for simplicity. But they have a lateral and a medial habenula. They have a telencephalon, tectum, and other structures. All the major neuromodulators are largely conserved as well. Now, how to image? We've worked as hard as anybody over the past few years in trying to design methods by which one can capture at high speeds cellular resolution activity in behaving animals. And these fall into a number of categories. In 2013, together with Logan Groslick and Michael Broxton and others in the lab, along with Mark Lavoie and Mark Horowitz, we developed a light field imaging method. We've also worked hard on advancing light sheet methods. I won't go into the technical details, but these have certain trade-offs between speed and resolution. We've also worked hard on two-photon methods where one moves a plane of illumination as rapidly as possible through the volume. Now, you can see this, speed resolution trade-off, and despite all the effort that we put into these methods, both light sheet and light field, in the end, I was dissatisfied with this core criterion we set for ourselves, was to make sure that even in dense clusters of cells that we had true, unmistakable, certain single cell resolution, and I wanted to maintain that. Here's an example of light field technology at its best. This is work collected by, this is data collected by Aaron Engelman, and you can see it's nice, but and you certainly can, where cells are isolated, see individual cells, but in dense clusters, it's hard to distinguish individual cells. So we, despite all the work, we ended up rejecting this as a method. Likewise, light sheet, which has a similar issue, although in sparsely labeled regions, you certainly can see and distinguish individual cells in dense clusters. You cannot. And people who are working with this method, including us, but others as well, will typically work with voxels rather than cells as their core element in analyzing their data sets, which has its own value, but we wanted to stick to cells as well. And I'm glad we did, as I'll show you later. Still, the light field method, for example, was interesting, and we used it to screen early on. We had to put animals through a protocol collecting baseline fluorescence activity using genetically encoded calcium indicators that reveal by their fluorescence the activity of the individual neurons. And we put animals through this challenge, this shock protocol, and then this set of experiments, we then embed the animals in augurus in a head-fixed fashion and image their brain to see if anything is different. And we saw something amazing. One structure in this setting showed elevated activity, prominently elevated activity, and it was the lateral habenula. So this was fish that had gone through this stressor and were in a passive state, and elevated activity is seen in one structure and one structure only, the lateral habenula, and that was blocked by ketamine, as the behavioral effects also were. So that was interesting, very interesting, and we wanted to follow this up. We wanted to get to single cell resolution, and so we ended up going to good old two-photon microscopy, but juiced up as well as we could with very fast scanning of a piezo through different volumes to let us capture the activity of essentially all the cells in the forebrain. We can get about 1 to 2 hertz captured of the volumes. The fish is embedded in agarose by its head, but its tail is free, so it still reports to us whether it wants to move or not, its level of activity or passivity by its tail movement. And this is the sort of data that we get. You can see even in dense clusters, we are quite confident of single cell resolution. This is what we wanted. And this has, and why do we want that? This is a capability that we're still capitalizing on, but that lets us do the following. In behavior, we can see the activity of individual cells, and then we, using the genetic encoded calcium indicators, GCAMs, we can then fix the cells and see the fluorescence maintained of those fluorescent molecules. There's some shape changes and size changes that happen when you fix a tissue, of course. And if you look at the live and fixed case in green and red, they don't overlap perfectly even in the same animal. You can see plenty of cases where it's not yellow, but red and green, separate, indicating that registration is not at all what you'd like. But Matt Love at Baron and Josh Jennings in the lab have worked hard on developing registration methods, and so now we can, at single cell resolution, work the live and dead and get them to align with single cell resolution. So then you can do rich staining for neuromodulators and cell typology markers in general, and refer that back to activity with cellular resolution and with the totality of the fish system during behavior. This work is still ongoing, but that's the reason for maintaining single cell resolution. And the animals certainly exhibit this passivity even in this head fixed to photon state, and that is not seen in the presence of ketamine. And so here's what the data, brain cell activity data actually look like. It's actually happening to the fish, happening before your eyes, the transition from active to passive. And you can see there's initially sparse activity across the brain, and then you start to see accumulation here at these two side structures of activity of individual neurons. And the movie will go through the entire epoch, and then it'll loop back and play again. You can watch it happen again. So here's back at the initial state. And this is an animal that's exhibiting behaviorally, we know, the transition from active to passive. And this accumulation you're seeing is very interesting. It's not as if the cells are going through calcium death and are dying and being pegged at a high level of activity. They maintain, in large part, dynamics. They also seem to make that transition at different times, some late. And this turned out to be very interesting, and another reason why we were glad that we maintained the single cell resolution. But all the way across the brain, the only structure that went up, and this was concordant with the light field data, this 2-photon data, revealed only in these lateral habenule structures did that transition happen, and it was blocked by ketamine, which is shown in magenta. Now, also very interestingly, there was an opposite direction effect in the raphe. There was a smaller but significant reduction in activity in the dorsal raphe, the serotonergic structures. And so this opposite valence effect was intriguing because if you remember the two projections from prefrontal to the habenule or dorsal raphe, the raphe had opposite direction effects on the transition from active to passive coping. So we were very intrigued to see that. But then we could look everywhere in the brain. We saw some many structures did not change. Nothing in the telencephalon. Now remember, fish don't have a neocortex, so this isn't quite the same as the neocortex. In mammals, I think a lot of the context and modulators and modifiers of these sorts of transitions that bring a lot of cognitive power to bear may help guide these transitions in the fish. That's not happening, at least not that we can see in terms of activity patterns in the telencephalon. Of course, there may be something more subtle going on that we don't understand. Also, dorsal thalamus showed a raphe-like effect. Many regions showed no effects. This is not we were somewhat reassured by this, that this stressful protocol is having relatively specific effects on specific parts of the brain, and that mapped somewhat clearly back onto what had been seen in the rats. There also was some interesting correlations, not yet causality in terms of activity, and behavior by interesting correlations. And here we looked at the persistence of the state of the animals. There was a baseline period, free swimming exposure to the inescapable shock protocol, followed by a 3-hour recovery period, long enough for protein synthesis to matter perhaps. And then 2-photon imaging in the head fixed state where we re-expose the animal to the aversive stimulus. And you can see in terms of both movements of the animal, behavioral action, and lateral habenula activity pattern, that the re-exposed animals in light blue exhibited a larger and faster transition here to the passive state and here to elevated lateral habenula activity. In fact, there was some correlation at the single fish level. Fish that had a larger lateral habenula activity change exhibited the greatest transition to passivity. So interesting, suggestive of causality in terms of activity, but not yet shown. And for causality, we turned to optogenetics. And in this case, we were able to use genetic strategies to target channelrhodopsin expression to lateral habenula structures. Talia Lerner in my lab did patch clamp recordings from the fish to verify that there was indeed light-activated currents, and there were activated currents in these cells. And then we looked at the behavior and brain-wide imaging. And so here's the behavior first. So if you look in just at the black trace first, these are channelrhodopsin negative fish. So these are not expected to exhibit a optogenetic effect. And when you expose these naive non-channelrhodopsin expressing fish to light, there's an increase in activity that's reversible. So there's a natural light response, elevated activity. Animals that are expressing channelrhodopsin in lateral habenula don't show that. In fact, there's a slight decrease that's reversible. Very modest effect in the naive fish, but in fish that have been previously exposed to the shock period, again, if you don't have channelrhodopsin, you have this little blip upward in activity that's reversible, but powerful suppression of behavior, of free-swimming locomotor behavior in these animals. And so this showed there was a experience-dependent, aversive experience-dependent causal effect of habenula activity on passivity. And then we also had this opportunity to, while we were stimulating the habenula cells, to look brain-wide at all the activity patterns of all the cells across the brain. So that was a really exciting possibility as well. And we've analyzed this. These are extremely large data sets, of course. You've got many tens of thousands of neurons and time series going on for many tens of minutes. And so we're getting good at handling it, but this is something that nobody's really solved fully how to address this. And I'm sure there's beauty and complexity within these data sets that we don't yet appreciate. But some things did pop out to our primitive primate eyes, and some of that is shown here. So here are fish now where we're co-expressing the channelrhodopsin just in lateral habenula, the GCAMP, the activity indicator, across the entire brain. And a reassuring thing is that if you stimulate the lateral habenula, you see elevated activity in the lateral habenula, so that's good. That suggests we're doing what we think we're doing, and it's fast. There's very little else going on early across the brain, at least that we can pick up here, where everything's normalized to what happens in the channelrhodopsin negative animals. So we see very little effect, it's laterally or contralaterally. But 8 to 10 seconds later, there's a prominent decrease in raphe activity. We think, based on this, that the habenula can be causally upstream raphe and driving this decrease that's seen naturally. And this is just the first step in our analysis of these brain-wide data sets. There's undoubtedly a much greater beauty and complexity yet to be appreciated. But nothing I've shown you yet fully capitalizes on the single cell resolution. And that's where I think even the most amazing thing came. I'll show you that next. So this is the same movie as before. And what I alluded to previously was that some cells seem to turn on early, some cells seem to turn on late. And so we looked at this systematically, looking at the latency. And here's an example of a late transitioning habenula cell. And here's a more early cell, and then some are more in the middle. And so we were intrigued by this, and first we asked, is there any spatial structure to these cells? And we looked at we color-coded here just for visualization, the early versus late transitioners. And if you make a projection, you can maybe see some clustering of cells with similar colors, and indeed we could identify that there was a statistically significant effect, that cells were more like their 10 nearest neighbors in the Hybenial in terms of the time when they transitioned than 10 randomly selected cells. But it was not an extremely powerful effect. We still are interested in exploring this further and determining if there is a typology or projection specificity to those cells. But what was most interesting was the following, is that there's a complete tiling at the single cell level, temporal tiling of the behavioral challenge period. Different cells carrying out the transition at different times in lateral Hybenial. That's not seen in Raphic. Okay, the cells carry out their transitions all at about the same time, and they don't tile the challenge space temporally, as it were. And so what it seems to us is that the Hybenial is set up to assess the temporal statistics of the challenge. And this is exactly what you want, to have an appropriately suitable behavioral state transition from active to passive. If there's a temporal statistic of the challenge that suggests that it's going to be ongoing for a long time, it may not be of value to exert a powerful exertion of resources in the present to escape it. And this accumulation of evidence, if you will, of the statistics of the challenge is something that Hybenial seems to be able to do. Other structures can't. We know the Hybenial is upstream, or can be upstream causally of the Raphe. And in an animal without a cortex, it seems that a lot of the assessment of the situation does fall on the Hybenial. We don't, of course, there may be other structures upstream that are carrying out the assessment that we can't pick up due to the limitations of our analysis. But one thing we know we're not missing is we're not looking at a particular brain region. We are collecting all the data across the brain. So we've taken a step forward, I think, in understanding where this computation might arise. Now, of course, in mammals with a cortex, that carries a great deal more cognitive firepower, and this is something that is well suited to evolve later and to modulate this behavioral state transition. But larval fish can do it with just their Hybenial. Now, as I mentioned, we want to delve into more detail on these individual cells that are carrying out the transition at different times. Here, this might have been one of the movies Matt was referring to earlier, but the ability to not just look at the individual molecular phenotypes of the cells, but then to assess their local and global wiring in the same organism is of value. And here's where we're starting to bring our hydrogel tissue chemistry approaches, including clarity, to bear. And here you can, in the same intact system, you can assess wiring as well. There's some advantages to this hydrogel tissue hybrid method, which we've been working on with a number of very talented students and postdocs in the lab over a number of years. Even back in 2013, we knew a number of key features of this method that are very relevant to this question. We designed it to be all aqueous based, so it doesn't quench or destroy the fluorescence of the native fluorophores, in this case the GCAMs. And so you could have even multiple different, you could have a GCAMP and an RCAMP, and the native fluorescence would be maintained. And the cell filling, axon filling quality of those would be maintained as well, and could be assessed in the same animal. You can go through many rounds of labeling. This would, in principle, let you get deep molecular phenotyping of the cells of interest. As we showed in 2013 and 2014, there are size changes that are reversible that can happen with the hydrogel tissue hybrids, and you can do both protein and nucleic acid labeling, so you can do in situ hybridization as well. So this is work that's ongoing in terms of assessing the nature of these early versus late transitioning cells. So that's the first part of what I wanted to talk about. It's the fish work. As you can tell, we're excited about it. It's a big chunk of the lab is working on the fish now. We, of course, still work on our rats and mice, and I think there's some value in working on both together. What I'd like to talk about now is the microbial opsins themselves. These are beautiful, timeless proteins. They have their own significance, of course, to the algae and to biophysics, and I just like them a lot. My deepest roots were in biochemistry. When I was undergraduate, I focused on my major was biochemical sciences. That's what they call it at Harvard. And it was, but I didn't have I'm in an engineering department, but I don't have an engineering degree. I have a biochemistry degree, and this is something that it's hard to shake, even though I've become sort of a systems neuroscientist. I guess I still mostly care about proteins. And these proteins come from these microorganisms, and here UCSF has a key foundational role here. Walter Stachenius and Deisseroth helped here at UCSF found the very first evidence for microbial rhodopsins, which was an amazing thing at the time. The vertebrate rhodopsins were known, but the microbes have them was a very striking finding. And they identified the bacterial rhodopsin, which is a proton pump. There were halo rhodopsins found 6 years later. That pump chloride, channel rhodopsins identified in 2002 are similar, but actually open a pore that allows ions to flow. The pumps carry out a discontinuous handoff of ions across the membrane. The ion coming in one end is not the same as the one that comes out the other end, and one ion is moved per photon. The channels are different in that they physically open a hole, and ions can pass through hundreds per photon. But they're all homologous to each other. They all use all trans-retinal as the chromophore. But this, when we wrote this review, Feng Zhang in the lab was now much more famous, but he was a grad student in the lab in the chemistry department, and he was absolutely crucial in all the optogenetic work. This was a review we wrote in 2011 before we had the crystal structure, and this was the level of understanding. Very interesting. There was a lot of controversy in the field as to whether there actually was a pore. Some people said it was a leaky pump instead. And then there was a question of where, if it were a channel, where the channel would be. There was not a clear precedent for a light-activated pore, semi-transmembrane light-activated pore, certainly. And the dominant theory, and there was even a cryo-EM structure where it was put forward as the most likely conclusion, was that there was not a pore within the monomer, but rather at the interface of a dimer, and that the pore would somehow be formed at the interface of one monomer and another. Now, we really, both because we like these proteins and because we wanted to change the properties of the protein, we were very motivated to get a high-resolution structure. And we worked very hard on this in collaboration with Osamu Nareki at the University of Tokyo. Shara Ramakrishnan in my lab made hundreds of different constructs. These don't express very well, and they exist in a variety of different photocycle states, so it's very hard to crystallize. But we finally got there in 2012. And I'll tell you, even before we got there, we were able to modify based on homologies. We were able to create improved versions based simply on structural homologies with the pump opsins. But nothing that we did could address the pore question. What is the actual channel pore like? We did other things as well, and Lisa was absolutely crucial in this work. Homology modeling allowed us to address a speed issue that the channel rhodopsins had. So if you look at the time scale here, 50 milliseconds, sometimes we would get doublets or triplets of spikes instead of a single spike, and we attributed this to a somewhat slow deactivation after light off. And we thought if we could speed up the deactivation after light off, we might address this fidelity issue. And so with some mutations, this was 2010 before we had the crystal structure based on homology modeling to the pumps, we were able to find some mutations, including this E123T mutation, or that we call a cheetah mutation, that greatly accelerated the deactivation and allowed us to speed things up. We also were able to greatly slow things down, and this was work with Andre and Lisa over Uzar in the lab, and much of this in collaboration with Peter Hagemann. What we found was that we could take this very fast deactivation here at 10 milliseconds after light off, and we could greatly slow it down to many minutes, even many tens of minutes. And that was with mutations in the cysteine 128 or the aspartate 156 or both together. And this created a bistable opsin, and these turned out to be very useful. And I'll come back to that and telling you about how they were relevant for studying anhedonia later. Now, you can still turn off these channels with a red-shifted photon, green or yellow or red. This is a simplified photocycle, a dark closed state of the channel that absorbs a 470 nanometer blue photon. And they end up getting stuck in this open state, but you can shortcut it back to the closed state with a green or red or yellow photon. So useful in terms of kinetics, changing the speed, faster or slower. But we knew nothing about the pore, and so we couldn't change the selectivity of the pore. We didn't even know where the pore was, or if it was within a monomer or at the interface of two monomers. But in 2012, we got to the crystal structure. It is indeed a dimer, but there's not a pore at the interface. Each monomer has its own pore. This is the all-trans retinal magenta here, and if you overlay this structure with other microbial opsins, there are two helices that are tilted in such a way that it creates a larger space, and you can actually see the inner lining of the pore as well. Many polar residues. It's a closed state structure, and we can see two constrictions, but a very clear channel pathway. No bound ions, no high affinity binding sites that we could see, and relatively though preponderance of predicted negative residues in terms of surface electrostatics, and the naturally occurring channel opsins at the time were all cation channels, non-selective cation channels. And so this suggested a possible mechanism that could contribute to cation selectivity. And so to test that, of course, we would have to really change selectivity. And you can imagine this is a tall order to really drastically modify the inner lining of the channel while trying to maintain this gating. The all-trans retinal is right here and intimately associated, and some residues even play key roles both in pore structure and in the setting the spectral and kinetic properties of the retinal chromophore. But Andre Barondes and Sue Lee in the lab were not afraid of this challenge, and so they expressed opsins, various mutants, hundreds of different mutants in cultured neurons. Not all the neurons look as pretty as this is a particularly pretty one, but they worked in neurons to make sure that whatever they came up with would be useful in the end. And their readout all the way through whole cell patch clamps, a low throughput assay, but this gave an incredible amount of value in understanding the reversal potential, looking for a signature of anion conductance, chloride conductance, and also assessing the health of the cells as well. And eventually they got there. So they found that they could, with after looking through hundreds of different mutations and making combinations of them, that they could take the inner lining from largely negative, as shown in red, to largely positive, as shown in blue. This is just predicted. We don't actually know, but just looking at the residues that are there and doing some simple modeling. And this was back-to-back with a very nice paper from Peter Hagemann, a similar result, different mutations also affecting the net internal electrostatics that would be predicted. And these were indeed chloride channels finally. And so we could shut off spiking with blue light instead of driving spiking. Another thing that was, we wanted to understand the channels. We wanted to change their selectivity to create inhibitory channels, which would be more potent than single ion moving pumps. But also we wanted to create bistable inhibition. And this bistability of getting temporally precise but prolonged inhibition, so you don't need to keep delivering light, was something many people wanted to do. But with a pump, you can't create a bistable state. There's no open state to stick it in. Only a channel can do that. And so we then brought the step function or bistable mutants to bear on this inhibitory opsin. And that worked. And so we could have bistable inhibition as well, induced by blue light and reversible by red light. In both cases here, we're injecting current to drive spiking. We call this switcher for step waveform inhibitory channel rhodopsin. And then a really amazing thing happened about a year after we described those results. John Sputich's group in Texas found a naturally occurring chloride-conducting channel rhodopsin. And we still don't know why a algal cell wants fast light-activated chloride flux. It's still one of a mystery. But it clearly does. It's evolved a beautiful one. And when John published this, we were very excited to go and see what principle we might find from its predicted structure overlaying its primary sequence on our channel rhodopsin, cation conducting channel rhodopsin structure. And what was amazing was they were nearly identical in terms of the predicted electrostatic potential at the surface of the inner lining. This was our described structure in 2014. This is what John Sputich published in 2015. And this told us a couple things, but most importantly that we have a decent understanding of the selectivity mechanism likely of the channel rhodopsin core. Also, you can't beat nature. Nature always gets there first and probably does it better. But beautiful work from Elena Goverenova and John Sputich. You can change other aspects of the other properties of channel function. Fong also found in 2008 the first red-shifted red light recruitable channel rhodopsin from this multicellular green algae, Volvox carteri, and that went through a number of iterations, including building on some work from Roger Chen's lab. He took some aspects of the Volvox backbone and made a red-shifted opsin called Reacher. And we sped that up with the cheetah mutations, and that is a very nice red activated opsin. And I won't go through all the work, but even back in 2011, you could get very fast red light driven spiking. All the opsins have significant recruitment in the blue even today, and so this is a source of some frustration. And we initially were very interested in seeing if we could do in vivo combinatorial control, drive one population completely alone, or another population completely alone, or them together. That's still not quite possible because of this blue overlap. But a much more important value for these red-shifted opsins became clear only later with the advent of the GCAMs. And the best indicators, the best calcium indicators are all blue light activated. So having a red control axis for the blue readout allowed all optical experiments. And this, just over the last year, allowed us to do something that we wanted to do for years and years and years in optogenetics, but never been able to do, which is to align the delivered activity with naturally occurring patterns, timing and magnitude from the very same animal, same task. And this finally was now possible. And this is an example of that. Tina Kim and Sam Yang described this last year, building on this fiber photometry method, which Lisa and Logan and Isaac and a few others in the lab developed and described in 2014. And it's a powerful method because you can administer deliver a single fiber that can be chronically implanted that both delivers and collects light. It's not forming an image, so we call it photometry, fiber photometry. But and actually that's a value. If you have a single time-varying scaler that's easy to work with, you can do, as I'll show you, closed loop work, very fast closing of the loop that lets you adjust what you're delivering to what's naturally occurring. And here, Tina Kim co-expressed a blue light activated genetically encoded calcium indicator with a red light opsin, a Volvox derived opsin, in the dopamine neurons of the ventral tegmental area. And so then she asked, well, what's the naturally occurring pattern of activity in response to reward in a freely moving awake mouse? And so she recorded that, giving these water deprived mice a little bit of sugar and water, and that's the naturally occurring pattern. And then she comes in and tries to mimic that. Coming in too hot here first, but she's able to back that off, and she matches the naturally occurring pattern. So it seems simple, but kind of a holy grail for a long time in the field, and now we can deliver and match in time and amplitude naturally occurring activity patterns. That's at the population level. You can do it at the single cell level as well. We and others have used spatial light modulators, basically holograms of light, to play in activity to individual single cells. And this movie, which I think is not showing up too well here, shows that we can control individual cells within visual cortex of an awake animal, again with this all optical two-channel control. So this is what you might call modern optogenetics. You can be acute or chronic, you can excite or inhibit, you can have this photometry approach lets you have multiple fibers across the brain, so you can have a global imposition or modification of a brain-wide state, and you can match or modify the time or amplitude of naturally occurring patterns. And this is the closed loop work that I mentioned. Vikas, back in 2009, did the very first optogenetic closing of the loop, but with an electrical readout and an optical control. And here, we now have an all optical closing of the loop. Most optogenetics still done open loop. You have some idea of what your system is going to require, and you can deliver that. But there's no fast feedback like the car cruise control will have. In closed loop, you can decide what level you want, and you can tune what you're delivering to match that and maintain it in real time. And Noah Young and Tina Kim have finally done this. So this is a photometry-based dopamine neuron closed loop experiment. They're collecting the dopamine neuron activity in this gray period, and then they choose that as the dotted line shows is what they're going to fix fix the activity out with their closed loop paradigm. The system's free running in this white period, and then they turn on their PID closed loop control algorithm here, and you can see they're able to lock the activity to this naturally occurring pattern in real time. Okay, so the last few slides then, I'm going to bring this back to this initial question of anhedonia and show you how the deeper understanding of these proteins themselves has allowed us to make headway on these difficult questions. And this was work led by Emily Barondes in the lab. She is a graduate student now doing her neurology residency at MGH in Boston. And she wanted to understand the brain-wide anhedonic state. She wanted to understand what is the, just as in the fish, we had a brain-wide readout of the passivity state. She was interested in the anhedonic state and its representation and its causality. And because we can't do whole brain real-time work in mammals, she took a Fmri approach, and optogenetic Fmri, in fact. This was an approach that had been pioneered by Jin Lee and Remy Durand in the lab back in 2010. That was in anesthetized rats, and what Emily's innovation was making this work with awake rats and in animals where there were behavioral states that were known to be present in the animals, and collecting causal information on how those states were represented. And the challenge is it's hard enough to keep a human being still on a scanner. A rat, it's very hard to explain to them the value of the science and all that. Even if you head fix them, they still are able to wiggle and you get these X, Y, and Z deflections. But Emily's patience and genius was in figuring out a way to habituate the animals, and so she actually was able to get them with a number of days of habituation to remain still on the scanner. And the habituation involved both with the head fixation and simulated sound of the scanner. And so she could then get beautiful data in awake rats, for example, visual stimulation, which she always had as a control. She can see nice blood oxygenation level dependent signals in visual cortex, superior corpus, and visual thalamus, but not in other regions of the brain. And so then, how to study anhedonia? Is there something to test, a brain structure to go in and modify and to assess the anhedonic state? And Emily was inspired by the decades of work from Helen Mayberg, but others as well, who observationally, Helen's known for both observational and interventional work with BBS, but in this case, specifically thinking about her decades of observational work on depressed patients. And she had consistently found by a number of measures, including PET and other measures, elevated activity in the subgenual cingulate gyrus. And this spot had a number of interesting properties. It was more active in patients with depression, prominently so in patients with anhedonic symptoms, reversed and predicted reversal of symptoms with treatment. And so this is elevated activity, suggesting a correlating at least with a pathological level of anhedonia and other symptoms. The other interesting thing, another UCSF related person, Anto Banchi, back in 2013, had a very interesting Nature paper, Chen and Banchi, where they looked at cocaine-addicted rats. And they had a very interesting result. I was not involved in any of this work. Admirer of it, though. They did an optogenetic intervention in prefrontal cortex, and they found that they could, by exciting prefrontal cortex, they could suppress cocaine-seeking behavior in these rats. Very interesting, surprising result. So much so that a clinical study trial was done and published in late 2015. Optogenetically guided clinical trial in human beings, cocaine-addicted human beings, using transcranial magnetic stimulation as an intervention to, best guess, the most homologous structures in these human beings. There was an experimental group and a control group, and this is not a heavily placebo-dependent population, and there was a prominent suppression of cocaine-seeking behavior and cocaine use in these human beings in the experimental group, control group. So here you've got, again, elevated activity, prefrontal, suppressing reward-seeking here in this pathological state, and here suppressing, it seems, at least by correlation, naturally, pursuit of natural rewards as well. So this was enough for Emily to say, okay, let's go in and deliver an elevation of activity in medial prefrontal and see what happens. And here, she was able to build, this is a bit washed out, but this is a summary of a lot of our work, a lot of the mutagenesis we've done on a lot of the interlining of the core and the retinal binding pocket. And as you'll see, a lot of the individual mutations and a lot of our structure function analysis turned out to be crucial to get this experiment to work. And I'll show you those individually in detail. So the first thing she did was to generate a chronic, stable, moderate excitability shift in the prefrontal cortex. And so what she wanted to do in this case was use these modifications of aspartate 156 and cysteine 128 to create the stabilized step function opsin and give bistable excitability. And so here, this step function opsin, excitatory in virus, delivered to medial prefrontal. You can see the expression pattern by YFP. The bistability, blue light, elevated excitability is recorded by extracellular electrodes in prefrontal, reversed with yellow light. And then bold signal validation of this effect, you can see elevated bold signals, the SSFO expressing, but not the controls. And again, the bistability here is crucial because she can elevate activity and then put the animals in the scanner. She doesn't have to bring the lasers and all that into the scanner. So bistability crucial here, and she can have animals carry out complex behaviors, social behaviors, sucrose consumption behaviors, and so on to test for anhedonia without having any apparatus present at all. So a good choice, I think, to go with the bistable opsins. And so is there an anhedonic effect? There is. It's clearly detectable. Rodents are very highly motivated by sugar water, but amazingly, as motivated as they are, you can see moderate changes in their preference for sugar water or regular water, and this is interpreted as anhedonic-like behavior, and it's not complete, but it is reliably reported in the literature, and she was able to see that as a consequence of elevated activity with medial prefrontal cortex delivered with the step function opsin, a mild but stable and reversible reduced preference for sugar water over regular water. No change in total water consumption. Also, there was a reversibly reduced social interaction, but no change, which is normally very rewarding for mice, but not novel object interaction or locomotor changes. So she was able to see anhedonic-like changes that were induced with this medial prefrontal intervention. And so then finally she wanted to use her bold fMRI readout and this intervention to see what the altered anhedonic-like state actually is, how it's represented and how it might be causal. And so she wanted to image, she also wanted to see what happened to a known rewarded state. And here the question became, okay, there's the resting state of the animal, but then there's also the rewarded state. Is this intervention affecting the rewarded state as well? And here the question became, how can you really know in the scanner that you have an animal that's in a rewarded state, and how is that modified? And here we were able to use rats, tyrosine hydroxylase Cree-driver rats that Ilana Witten in the lab had made and described, targeting a channel opsin to the ventral tegmental area dopamine neurons and getting selective drive of dopamine neurons with a fiber optic implanted there. And how do we know this is rewarding to the animals? They will press levers many hundreds of times per day in order to get laser light delivered to these ventral tegmental area dopamine neurons. So they will work very hard for light to these neurons. And so we know it's a rewarded state. By the way, this was interesting in its own regard. There was some question as to what, there's a vast human literature on gold imaging in various states of reward, but it was unclear if this state was causally related to dopamine neuron activity in the VTA. This study showed that that was the case. We could see, in fact, very specific activity in dorsal striatum, ventral striatum, and retrospinal cortex. And the correlation between ventral striatum bulb in particular and the number of lever presses an animal would carry out was significant. And so the level of recruitment of this activity pattern, VTA driving ventral striatum, which includes nucleus accumbens recruitment, seemed to be an important part of the rewarded state. Okay, so then Emily's question became, what happens when we elevate activity in prefrontal cortex? Does that affect this rewarded state? And if so, how? And so here, using not just the step function opsin with these modifications, but the Volvox-derived opsins C1V1 for red light control. So we've got a blue light bistable opsin in prefrontal cortex, and then she's going to ping the VTA dopamine neurons with this red light activated opsin. And so this is a pretty amazing experiment. I think you can see the two fiber optic tracks coming in here, prefrontal and going into the VTA. And also just the implications of it. She's asking, how does activity in one brain region affect conversation to other brain regions they're having with each other across the brain? How does prefrontal cortex affect the causal ability of VTA to recruit this ventral striatum accumbens activity? And she saw a very powerful effect. Here, just the baseline, again, similar to previously, the C1V1 recruitment of this striatal activity, dorsal and ventral, in the absence of the prefrontal elevated activity. And then here, elevating the prefrontal activity, she saw a powerful suppression of that recruitment. So, elevated activity in prefrontal affecting how these two other regions are conversing with each other. And that was interesting, not just in terms of the brain-wide physiology, but also was behaviorally consequential as well. There's a place preference task. You can get animals to very quickly accumulate the time they spend in one chamber of a two-chambered apparatus if they get a rewarding stimulus in that chamber. And what she found, if you get dopamine neuron activity in one chamber, you'll typically spend more time in that chamber, as the rats reported. But if you elevate prefrontal activity, you can see a modest but significant and reversible effect, reducing that preference for that chamber. So, in some sense, I don't know about delight, but certainly these are animals that are no longer pursuing what they formerly were pursuing as a result of this very specific intervention, and maybe a step toward understanding anhedonia. So coming back to Sam's poem now, without the dashes and capitalizations, to see it in its true glory. You can, I think it's actually good the brain's too dark inside. If it were any lighter, we wouldn't have been able to do it. And the actual, the beauty of these proteins themselves and the instruction that came from microbial introduced systems, I think it's something that can't be stated too strongly. So I want to thank everybody. I think I mentioned the key people along the way, but I'm just really lucky to work with these amazing people and so many people here as well in the room that's been a pleasure to work with and learn from. And it's been an honor to kick this off, and so thank you for the invitation. Thank you.
So we are going to run straight into a reception. Karl is going to remain around for the reception. So for folks who have a detailed question after that tour de force, you'll have lots of opportunity to be able to answer that. I just want to close by saying that Sam pointed out at the start of this that he came to UCSF in 1986 based on this very early and beautiful work beginning to move from really the black box of the cell to understanding the component parts and molecular dynamics, and then being able to jump all the way to behavior. And then realized when he got here that maybe his optimism was a little premature, and that there was a tremendous amount of work that needed to be done in order to get from there to understand the complex relationships between molecule, cell, circuit, ultimately complex human behavior and psychiatric illness. And what I'd say is the purpose of this lecture series is to reward Sam's optimism, to underscore that. And I think we've seen that in the incredible work of Dr. Deisseroth in building those links and providing us all in neuroscience the tools to now take the next step to disentangle that complexity. And I pledge to you that each year we will have another speaker who will underscore that your optimism was well-founded. Thanks, Sam.